So the last section here in our module on data distribution has to do with common distribution types. Okay, and we will start. So I have here a diagram that shows you the fact that there are many different types of distributions, but we're going to focus on these different distributions here because they're particularly important in biology and experimental science, and I have a few things to say about each one of them. So the first one is in a way fairly simple. It's a uniform distribution. That means that the probability for each range in your variable is the same. So I have an example here. In average, this is probably not entirely accurate, but in average, the probability that a child is born a different month of the year is more or less the same. Okay, so the number of kids born on different months is more or less the same. So you can see here in the graph, birth month, number of births. Okay, you'll have something similar if you throw a dice. Your probability of having a 3 or an 8 or 12 is going to be similar. If you flip a coin, your probability of heads and tails is similar. And so you have many processes in biology that on the basic level are uniform, equal probabilities of all the outcomes. If you have genes that that combine and split during meiosis, for example, you'll have equal probability that you get one of the two chromosomes versus another. Okay, and this concept of how it's important in biology will relate to the next distribution, the normal distribution. So I'm pretty sure you all heard about the normal distribution, a Gaussian distribution, and the idea that I want to stress here is that the normal distribution, the Gaussian distribution, is related in a way to a uniform distribution. And here's how. Let's take an example from biology. So again, let's take your genes. Let's say in a certain species, we figured out that the height or the length of the individuals are related only to 5 genes. Okay. And for each of these 5 genes, there's 6 alleles. So basically, if you get one allele, you're a little bit taller. If you get another, you're a little bit shorter. And let's simplify. Let's say it's a sort of simple, like 1, 2, 3 kind of scale. Okay, so you can relate. So the height or the length of the individual is going to be directly related to the mix you get from these 5 genes and 6 alleles. Okay. To simplify conceptually what I mean here, think about throwing some dice. Okay, so you have 5 dice, each has 6 faces, and so imagine that the height is related to what face it falls on. If you get all ones, the sum here is going to be 5, and you're probably going to be one of the shortest individual in the population. If you throw all this and you get all 6, well, you're going to be the tallest. Now, the probability that you get like 4 1s and 4 6 is fairly small. It's going to be hard to throw like that because to get something that's more in the middle here, you have many possible combinations, right? So to get 17 or 18, you can get a 2, a 5, a 4, a 1, and a 6. If you throw your dice one by one and you get a 6, a 1, a 4, a 5, and a 2, it's a different set of a different throw, right? But you get the same number. Whereas here, you need to get an exact sequence, and there's only one possible sequence to get those things. So, I mean, the fact is that each dice or each die has equal probability of getting each number. You sum those numbers, and you get a Gaussian distribution of the summed process. So going back to the example in biology, the heights will be Gaussianly distributed, even though the genes related to the height you might have uniform probability of getting one versus the other. So I thought this was a very important concept and very interesting. Basically, the fact that your normal distributions comes from the fact that you can add processes that are distributed. Actually, it's not just uniform distributions, but it's simpler to illustrate with uniform distributions. Adding many processes that have this random distribution, you can get a Gaussian distribution. Okay. The other thing I wanted to point out or the other couple of things I want to point out related to the normal distribution or the Gaussian distribution is, well, first, I'm showing you an equation here. It's not really for you to remember the equation. What I want to point out is that all the variables, all the things in there are constant numbers or known numbers. So E, for example, is 2.1 something, so it's a fixed number. Pi, it's a fixed number. All these things are known variables or pre-existing variables, except these two here, the mean and the standard deviation. And so the exact curve, the exact shape of your curve is completely defined by its mean and its standard deviation. And that's usually not the case for most of the other distributions that we have to deal with, like exponential, Poisson, you'll see some of them. And so it makes things easier because when you want to do math on something that is defined more simply, all the math becomes simpler. So that's one of the strengths or the big thing about the Gaussian distribution. Next thing I want to point out here is a couple of rule of thumbs, because if you have this distribution, its shape, as I said, very standard, all defined by the standard deviation and the mean. And so you can figure out how many of your samples fall within a certain distance of your mean. And you have all the numbers here, the cumulative percentage. But what I like to remember is these two rule of thumbs. So one is that 2/3 of your samples fall within one standard deviation of your mean. So this is 0 here, so this is your mean, let's say. This is one standard deviation here and here, and you can add these numbers, 15 plus 19 plus 19 plus 15. That is more or less equals to 2/3. It's actually 68.9 or something like that, but it doesn't matter, round it up just to have an idea. The bulk, about 2/3 of your samples are within one standard deviation. And then the next point here is that the vast majority of your samples are going to be within 3 standard deviations. So you see 3 here. You have 0.1% here that is in this tail, 0.1% in the other tail over there. So 99.8% of your samples are going to be within 3 standard deviations. Okay, very important to remember. Okay, let's move on to a few other sort of, wouldn't say more exotic, but less well-known distribution. This is a binomial distribution. It has to do also with random process, and so this idea of randomness is very important in biology and experimental science. And so relating different shapes to a certain random process is important. If you throw your coin and it falls either on heads or tails, right? Or you have offspring, like mice offsprings, they're either males or females, zeros or one. Okay, so you can draw your distribution here. If you have 2 births, average, like probably you're gonna have one that's male, one that's female. Okay, this is just an example. If you have 4 births here, how many do you expect? In how many cases do you expect to have? Sorry, if you have 2 births, how often do you expect to have 2 males? How often do you expect to have 1 male? How often do you expect to have 0 male? And so your probability is basically have to have one male because you can have female male or male female depending on the order, but if you have two males or two females, it's less probable. You can continue calculating the probabilities like this. If you have many samples, many repetitions here, this distribution will start looking a lot like a Gaussian normal distribution. But for fewer samples, it's very different, and so it's something to keep in mind. Poisson distribution. So the Poisson distribution is named after a French mathematician, and it relates also to this whole random process, and you will see in the tutorial how we can derive directly from a random uniformly distributed process. We can create events that are then distributed according to a Poisson distribution. And so here it applies very often to the number of these events across a certain like time window or space, space area. And so the example I have here are seeds that fall from a tree. So the seeds fall randomly, maybe, the level of how ripe they are or something. And so each have a certain probability of falling, and you measure at different distances from the tree. How many seeds do I have here versus there? Different distances here. And you'll get something that has this kind of shape. So Poisson distribution are sort of characterized by these tails. So in some instances, it can look nearly Gaussian. Now the instances, it looks something like you'll see the next distribution, an exponential distribution, but they're not exactly the same. And depending on this extra parameter that you have in the function, they will look more or less skewed on one side. And so stay tuned for the tutorial and the demonstration of how it's related. But bottom line, events and how they're distributed across a certain amount of time window or space plot. Okay. The exponential distribution, it's also related to intervals or to events, but in this case, it's the interval between events. So if you were, for example, to take coin flips and you're trying to figure out the interval, the number of flips between two successive tails, you'll get an exponential distribution. It's going to be the highest probability at the shortest interval, and then it's going to go down exponentially. If you take just a simple coin, the probability that you get a tail, let's say you flip, you get a tail. Okay. Now, next one, interval between this instance and the next one. Well, you flip again, half of your chances that you're going to get a tail. So you will get half the chance to get an interval that's that small. And you'll see that as the interval gets longer, you'll have a lower probability. So always highest probability in the first interval. If you want to have a tail that's one interval away, so you need to have heads and then tails. So probability of getting heads is 0.5 multiplied by the 0.5s for tails, and so you have 0.25, so you fall there, and then 0.125 you fall, and so on and so forth. So that's your exponential population. Again, there's a parameter depending on probabilities, like how likely those things are. Let's say it's not 0.5 chance of getting one versus the other. Well, the shape will be a little bit different. That's why we have several curves here. Okay, last category that I want to talk about is multimodal distribution. So that's not a specific function. It's sort of a general category. And I talk about it because it's important regarding the testing the shapes you'll see in a second. But multimodals basically mean several mode. So a unimodal distribution might be your Poisson distribution, might be your Gaussian distribution. You basically have one peak. Okay. If you have several peaks like you have here, this is a bimodal distribution. And it relates to this concept of mode, that's the most likely value in a distribution. So here you have a mode around height of 3, and here you have another mode. So if you look at the population as a whole, rigidly you have only one actual mode, but the term bimodal refers to having 2 peaks like this. So this is a bimodal distribution. You can have several other peaks, and then it's multimodal. This is important because often these distributions come from the fact that you have something that's maybe Gaussianly distributed, but you have 2 populations, 2 groups in there. So maybe height of this certain plant or something, you actually have 2 species of plants where you thought you had one, and one species is smaller than the other one. So they're both like Gaussianly distribution in height, but you had 2 groups, and so they look like this odd distribution. That's something that's important to notice, because a lot of statistical tests require a Gaussian distribution. So if you do a t-test and ANOVA, it works, it's accurate only if you have a Gaussian, a normal distribution. And so if you have something that's bimodal, you can't really run your parametric statistical test. So one of those sort of simple statistical tests like t-test or ANOVA. So often you need to test whether your population is normal or not. And there are statistical tests to do exactly that. You give your set of samples to this test, and it's going to determine if it follows a normal distribution or not. I want you to remember that name, because to be able to find it easily and replicate it. So Kolmogorov-Smirnov is the name of the test to determine if your population is normal or not. I have a little mnemotechnic trick. Smirnov, thinking about the vodka. If you have vodka, you're not quite normal. So testing the normality of your distribution, I associate it with this term here. And so I can recognize it, and I can sort of think back, Google it or something. Okay, so let's say you're in a bimodal case like this. Your data is not normally distributed, and you're like, oh, well, I wanted to use an ANOVA, but now I think I can't. And you realize, well, okay, it's bimodal. Maybe I have two groups. Let's say you measured the concentration of some kind of protein, and for some reason you have a group of healthy individual and a group that has a disease. Well, it's 2 different, and they have 2 different sort of mean protein concentration. And so if you put in that factor, you can sort of remove that average and ask whether your distribution is Gaussian around the mean of these 2 groups. So you sort of treat them separately, but your whole analysis can still rely on your whole data set. And you'll have 2 normally distributed population, and you can run your statistical test as long as you put in that factor, and you'll see when you run those things. Okay, so this is it for today. There's a few concepts here in this last part, so make sure you check them out and practice, test yourself with questions related to those. I will see you next in Module 3.